All right. Thank you so much for the nice introduction and for the invitation. I just realized yesterday I was checking my CV. It was my third brain map that I did. The first one was in 2010. And then I came back in 2015. And in that brain map, I actually introduced the first result of our study reporting brain inflammation in chronic pain patients. The first of our publications is kind of serving as a prologue to the story I'm going to tell you today about. So yeah, today I'm going to basically give you a little bit of an update of what we've been doing in the past 10 years or so. And as you can see, I'll focus a lot on brain inflammation and chronic pain, which, by the way, by now we're convinced is a phenomenon that is quite pervasive. Initially we had described it in chronic back pain. I'm going to show you that it seems to be present in many other conditions, etiologically different conditions. But also I'm going to venture a little bit beyond and outside of my comfort zone, below the neck, and show you a little bit of data suggesting that TSPO, which is this ligand that we've been using, many people used to image brain inflammation, is potentially also a marker of peripheral inflammation. So this suggests that this is a very versatile tool that could give us the possibility to investigate inflammation in the entire person, entire organism. But let's start from the top. So what is neuroinflammation? So neuroinflammation is an ill-defined, is a rather vague and in fact a little bit controversial nowadays term, which describes a series of molecular and cellular responses that are enacted by the nervous system when it is facing usually a threat. So that's the typical scenario. For instance, in the context of bacterial or viral infections, neurodegenerative processes, injuries, exposure to toxic metabolites, et cetera, et cetera. The key cellular players that contribute to neuroinflammatory responses are within the central nervous system, glial cells, so non-neural cells within the central nervous system, with specific focus on microglia and astrocytes. These are the cells that are more commonly used in investigating in this context, but also peripheral immune cells, circulating immune cells such as macrophages, monocytes, can contribute to a neuroinflammatory response, occasionally by crossing the blood-brain barrier or the blood-spinal cord barrier and penetrating the parenchyma, the brain and the spinal cord itself. Now, in the acute context, these responses have an adaptive value. They're good for you. They allow an organism to essentially identify, isolate, and clear a potentially harmful event and factor, a potential threat. But sometimes this neuroinflammatory response can become pathogenic in themselves. They become the problem because maybe they are disproportionately large, and they may be overstaying their welcome, meaning that when the original initiating factor, pathological event has been long gone, they continue. They do not subside. And so that's when they become a problem. And dysregulated neuroimmunity is being implicated in a larger and larger number of conditions that go from neurodegenerative disorder, from Alzheimer's to multiple sclerosis to ALS, etc., to psychiatric condition. There's a neuroinflammatory hypothesis of depression and many other conditions. Nicole studies its relationship with autism, etc. And as I'm hoping that my talk will show you, also potentially pain is one of those conditions that can be linked to dysregulated neuroinflammation. Why do we think that neuroinflammation might be relevant to pain? Well, this is not a new idea. There's been hundreds of studies using preclinical models, so animal models of pain, that have implicated glial cells in the establishment and or maintenance of persistent pain behaviors. And I'm going to focus on the animal aspect of it. So it's really the vast majority of studies come from the literature of using animal models. So we know that if you take an animal and you subject it to an injury, a pain-inducing injury, cells such as the glial cells, like microglia and astrocyte, in this case, in the dorsal horn of the spinal cord, this is where this image are taken from, but it's increasingly understood that everywhere else as well, in the central nervous system up to the brain, they're capable of detecting the presence of this threat, and they react. And they react by undergoing a series of cellular molecular responses, which mentioned before, which can include an increase in cell density, morphological changes in surface expression of markers, etc. And perhaps most importantly, synthesis and release of cytokine, chemokine, and other inflammatory mediators that sensitize the pain pathway. So now a simple brush with a innocuous brush, it used to be completely innocuous, to the animal becomes an excruciating experience. And this matches up what we see a lot in the clinics. Now, why is this exciting for us to have found out this? Is well, because if you modulate these responses, if you dampen neuroinflammation in animal models, well, the nociceptive behavior, so the animal pain behavior, are reduced or limited. So this suggests that neuroinflammation may be a target, right? We can use in our patients. Well, this is unfortunately where there's been a little bit of disconnect between the preclinical literature and humans. The evidence linking neuroinflammation to human pain is limited, and this is the big issue. I like to show this image here by Frida Kahlo, which I think is pretty interesting. This was a portrait, self-portrait she wrote, she painted in 1944. And you can see this was after she had a bus accident. She was actually traveling on her bus going back from school to home, and her bus actually collided with a trolley. And, she ended up with big injuries, and you can see how she describes it. You can see her, the spine basically feels like a broken column, which is the title of this portrait. And you can see how she describes the pins and needles that obviously look like they follow the dermatomal distribution of the sciatic nerve. I don't know if she would have conceptualized this way, but it was a beautiful way of showing how she was feeling this type of neuropathic pain. The pain was due to a damage to her nervous system. This was an aside, but going back to the concept we're trying to explain, so the evidence linking all the neuroinflammation work in animals to humans has been very limited, right? And what is the issue here? So the issue is that if you don't know if neuroinflammation even happened in a human with chronic pain, well, you don't know whether it's worth investing efforts and money to develop therapeutic strategies to address it. And you don't even know how to interpret clinical trial, including negative clinical trials. So you don't know if a supposed glial modulator didn't work because, well, neuroinflammation doesn't matter in pain, or maybe it's because it was the drug that didn't adequately target the intended target and engage into the target. Maybe the dose was not correct, etc. This is why my laboratory and other laboratories around the world working hard to try to devise methods to detect whether we can see inflammation in pain. How do we do it? How do people do it? I think there are three broad categories of studies that people can do to see if neuroinflammation happens in humans with pain. There are postmortem studies. There are analysis of the cerebrospinal fluid. CSF is the fluid that kind of bathes and flows inside and around the central nervous system. And of course, each of these studies have their own pros and cons. They can be very powerful, but at the same time, they are limited in their applicability. Of course, postmortem studies can, by definition, be only employed in deceased individual. CSF analysis, drawing CSF is not trivial. It's quite invasive, etc. So that's why my lab really focuses a lot on non-invasive imaging. And today I'm going to focus mostly on non-invasive imaging of a protein, which is called the translocator protein, or TSPO. What is TSPO? So TSPO is a 18 kilodalton 5-transmembrane domain protein, which is expressed on the outer mitochondrial membrane. It's not specific to microglia. This is something that we see over and over in the literature. It's expressed by different cell types, microglia, astrocytes, but also endothelial cells, some neurons, etc. So, and this has given a little bit of a bad rap to this protein, right? So how do we use it as a marker of a neuroinflammatory response if it's so, promiscuous, right? It's so expressed in so many different cell types. Well, this is because while it is true that it's expressed by many cell types as basal expression, many cell types, the level of expression in the healthy CNS is very, very low, becomes dramatically increased in the context of neuroinflammatory response. So this increase is driven by glial cells, immune cells. That's why. And this has been confirmed in many, many studies, including models of pain models. Another point that I would like to make is that recent postmortem literature in humans suggests that regional increase in TSPO signal might be mostly driven to an increase in glial cell density, which is something we want to keep in mind. So when we talk about TSPO PET signal increase, what we might be imaging is really the increase in glial cell density, which is a specific aspect of a neuroinflammatory response. And of course, as I've talked to you about an increase in TSPO PET signal, it's because we are able to image it using positron emission tomography. The idea itself of imaging TSPO as a marker of neuroinflammation is not new either. It has been, you probably have heard about this PK11195, which was the prototypical ligand. It was used to image many, many different conditions. The problem with PK11195 is that it has a low specific to non-specific binding ratio. What that means is that it goes and binds to TSPO, which is what we want to image, but also binds a little bit to other targets. Thankfully, now there is a second wave, and there's even a third generation, we can talk about it a little bit later, of TSPO radioligand that has higher specificity. For instance, PBR28, which is an 80-fold higher specific binding in primate brain, just to give you an idea of what that can allow us to do. Arguably now, we are in a position to image TSPO and, and neuroinflammation with a sensitivity and a specificity that we didn't have before. And by the way, PBR28 is, I believe that, I don't know if Gray can confirm, but I think it's still the workhorse at the Martinos Center. Yes, I see you saying yes. So there's a lot of interest in doing PBR at the Martinos Center. And these are a few, it's a sampling menu of the beautiful work that people like Caterina Maniero did in multiple sclerosis, then Arrosis Huntington disease, and then Nazem Atassi, and more recently Suma Babu in ALS. And I love to show this picture because essentially this image, the take-home message for this picture is that if you inject PBR28 in patients with neurodegenerative disorders, you'll see that the PET signal will be elevated more or less exactly where we expect to find pathology and where you know that you should be able to find inflammation. MS lesion, in Huntington disease, you see basal ganglia, which is a region that is very crucial in pathogenesis of Huntington disease. ALS, you see TSPO signal elevation in motor cortex, premotor cortex, cortical spinal tracts, with links to upper motor burden, motor neuron burden, cortical thinning, et cetera. So hopefully, it is my hope that by now I have convinced you that TSPO can be a powerful tool to image pathology and inflammation. But what happens if we inject now PBR28 or a TSPO ligand in patient with chronic pain condition? Do we see PET signal elevation, which is indicative of neuroinflammatory responses? And this is the paper that I was referring to before. This was our prologue. It was the first paper we published back in 2015. And we started with the chronic back pain patients, and you can see it was a pilot study. And you can see that immediately, this is a median image of chronic back pain patients and a median image of the control. You can immediately see how this part of the brain, which is the thalamus, show elevated PET signal, clearly elevated signal in the patient compared to controls. Of course, we did statistical analysis, and we were able to show, again, that thalamus is significantly different between the two group of patients, but also we saw additional spots, including the primary somatosensory cortex. We'll be back describing this data in a minute. But I think what was really exciting to us, to me at least, was the level of consistency of the signal. So if you actually take a cutout of this part of the brain, this axial view of the brain, and then you plot it for each patient and each control, and by the way, you can see there are these arrows that denote matching pairs. Each patient is matched to a healthy controls by means of sex, age, and a genetic polymorphism, which I'll talk to you about later. You are able to tell me who's the patient, who's the control, right? Without even just looking at statistics. And for us, this was quite exciting because, we don't have a biomarker for the presence of pain and such. So this was an interesting result for us. Of course, we were still dealing with a very tiny sample size. So the first thing we decided to do is, well, let's replicate it, right? Maybe just pure luck. So we enrolled new patients and new controls, and voila, we can still see the thalamus being hot in the patient, but not the control, and the group statistically significant there was a statistically significant difference over there. And by the way, one thing I want to mention is that in either of these studies, But also in none of the study I'm going to show you later on, we saw a sex effect. Now, in the, broadly speaking, the field of neurobiology, but in the field of pain neuroimmunology, the concept of the sexual dimorphism of the role of glial cells, and it's very hot right now. So it's pretty obvious that different cells have a different involvement in different sexes. So I know this question always come up. I'm going to just put it away right away to say that we didn't observe any sex effect either here or in other studies. So yeah, I was saying that, the thalamus, we were able to replicate. One thing we were not able to replicate. Oh, yes, you have a question. Yes. So the question was, I was wondering if the patient were using any drugs. Yes, yes. So we had some limitation about, for instance, the amount of, opioids people were allowed, but everybody was certainly taking medication, or many people take medication. We our post hoc analysis reveal that suggest that any taking drugs would be responsible for signal increases, because most people do take the drugs, but not everybody. Yet we saw this signal that was quite consistent. But that was a very good question. I can tell you that, a follow-up study, I was not going to talk about it, is actually to try to see whether exposure to chronic opiates can actually lead in itself to development of inflammation. And this is a study we are actually doing with Everati in patients with HIV neuropathic pain on opiates. So it's a very good question. Any other question at this point? All right, so I was saying, excitedly, we were able to replicate the result in the thalamus. But then, remember before I was showing you, let me just go back here. We saw other spots, including the primary somatosensory cortex. We didn't see that signal in the replication data set. So what happened? Well, one possibility is that, that observation was spurious. Yeah, again, small sample size, it's possible that, there was like a chance observation. But the other possibility was that maybe there are some differences between these two studies and might explain the difference in the cortical signal and the S1 signal. And as we were thinking about it, we realized that there was some potential difference in the clinical presentation of the patients. In the first study, all the back pain patients have back pain, but also some amount of leg involvement. So some symptoms going down the leg, okay? In the replication data set, we have this patient, but also some patient had only axial pain, so pain in the back, up to the buttocks, right? So maybe that's what it is, right? So what we did, we tested this hypothesis, we compare axial and radicular patients. So patients with again, axial only pain or patient with radicular pain is the pain that goes down the leg, right? In a dermatomal fashion. And the thalamic signal were unable to distinguish the clinical presentation of these two subgroups, but we were able to indeed show that patient with radiculopathy with the leg pain had a higher engagement of the primary somatosensory cortex. So, okay, so maybe it's the present radiculopathy that induces this, enhanced engagement of the cortex. But then we realized that we look closely that there was an additional difference between these two subgroups, in that the patient with radicular back pain also scored higher on the measure of nociplastic pain. So I'm going to put a pause here because I'm assuming that many of you will have heard this word for the first time. So nociplastic pain is a term that was recently introduced by the International Association of the Study of Pain, essentially the third descriptor to separate this kind of pain from the other two kinds of pain. So one is nociceptive pain, which is the kind of pain you have if there is ongoing inflammation in your tissue or actual damage in your tissue. Think about knee osteoarthritis. I know that my colleague Mohammed Jaraya will remind me that not all knee osteoarthritis is only nociplastic, but not only nociceptive, and I agree with that. But largely, I would say we would expect in most cases the type of pain where you have like a bone on bone and complete degeneration of the cartilage is really driven by the peripheral tissue damage or inflammation. If instead you have a lesion to the nervous system, right? Let's say you have, a soldier that had, a bullet that pierces maybe sever a nerve. Well, the type of pain is going to be neuropathic. Now, we realized recently that we need probably a third descriptor, which is that we describe the kind of pain that is not visible. It's not thought to be induced by visible changes in the periphery, there was changes in the afferent pathway, and that's the so-called nociplastic pain. So it's the pain that is thought to be due to the aberrant alteration in the central nervous system itself. And the prototypical condition for that exhibits this type of pain is fibromyalgia. Took me a little while to go to this little detour, but I think it was worth it. So essentially, we hypothesized that was nociplastic pain, not the presence of radiculopathy that was actually driving the signal in S1. And indeed, this was confirmed by correlation analysis. We saw that the S1 showed an elevated PET signal in patients that had higher nociplastic pain. And to me, even more exciting was that this was replicated by a different group. This is Paul Hodges in University of Queensland in Australia. They scanned with a different TSPO radioligand, but a TSPO radioligand nonetheless, patients with back pain. They separated them based on the phenotyping in patients with more nociplastic pain features and more nociceptive features. And they were able to show that in the primary sonosensory cortex, indeed, there was a difference. There was higher signal in the patient with nociplastic pain compared to the nociceptive and the pain-free individuals. Another theme that is emerging throughout all the studies we've been conducting is that the clinical significance of the TSPO PET signal appears to be regionally specific or regionally dependent. It's not that everywhere you see elevation, that's pain. Maybe it's pain in S1, but maybe it's something else in a different region. And indeed, we tested this hypothesis by focusing in this case on depressive symptoms. As you know, chronic pain is non-surprisingly linked with a high depression and anxiety comorbid to pain. And what we're able to show is that in region that just makes sense from a standpoint of the functional anatomy we are aware of, the pregenual SCC, the anterior medial cingulate cortex, the higher the back depression inventory scores, the higher essentially the severity of the depressive symptoms, the higher the PBR PET signal. And what we are able to, so I think this is interesting because while, in the slide I showed before, I was excited about other people replicating our work. Well, I think this is a little bit of a replication of other people's work because in the context of, we were able to replicate in the context of depression comorbid to chronic pain, the results from the literature of the major depression disorder. So in other studies, other people have shown that in patients with major depressive disorders, the higher the PET signal, more or less in the same regions, the higher the depression. And this data supports a neuroinflammatory hypothesis of depression. Now, one thing that I failed to mention is that this work was done not only in a PET scanner, but in a PET MRI scanner. Some of you may recognize this picture, courtesy of Cyprian Katana. This is our beloved Bay 6, who is, I don't know, how is he doing right now? Cyprian, is he doing okay?
It's up and running.
It's up and running, fantastic. And so this was the very first PET MR system that we had in the nation, and we were lucky to have it here. And again, this was a PET MR system, meaning that you can do simultaneously PET and MRI. So while we're acquiring all the PET data that I was showing you, we're also acquiring resting state fMRI data. We're acquiring diffusion MRI. We're acquiring spectroscopy, etc. And so this allows us to try to investigate the association between the PET signal and measure brain health and brain function. So for instance, we're able to show that, again, in our back pain patients, in the PET signal in S1 was not only proportional to the degree of nociplastic pain, as mentioned before, but also to the degree, to the strength of connectivity between the thalamus and the S1 itself. And in this paper, this one here, where we're showing you before, documented the link between TSPO signal and negative affect, we saw that the pregenual ACC PET signal was not only proportional to the back depression inventory score, but also to the strength of connectivity between cingulate cortex of pregenual ACC and the dorsal prefrontal cortex. And this, frontocingular connectivity pattern has been heavily implicated in the depression literature. Now, this is, of course, now the question some of you may have is, okay, what is the chicken, what is the egg, right? Because is it the changes in connectivity that drives the the change in PET signal or vice versa. And we did try to address these questions by running some statistical models. For instance, using this mediational model, we were able to show that the inflammatory signal in the pregenual ACC mediated the association between the frontocingular connectivity and back depression inventory. So you can start interrogating this question. I think more work needs to be done to really understand the causal relationship between these two measures. But at least it gives you a bit of a sense of the type of things we can do with integrated PET-MR. So this is a more recent paper that we published in Brain Behavior and Immunity. And in this case, my former postdoc Ludovica Brusaferro, who's now a faculty in London, at London South Bank University, she was actually interested in investigating the effect of the pandemic, living in the pandemic in our brain health. So, she basically compared healthy volunteers, but also chronic back pain patients, scanned before and after the onset of the pandemic. And she was able to see, which is a little scary, if you will, that after the pandemic, on average, people show more evidence of TSPO PET signal elevation, more inflammation, which we think is some kind of a stress response. Why do we think that? Well, because we saw it also in patients that didn't have a history of COVID-19. So it's not really the effect of the exposure itself. Among the many things that she looked at, also she was wondering, is there also an effect on brain age? Is it possible that people that have been scanned after the onset of the pandemic, and we live through lockdowns and worry about the future, etc., it's possible that our brain looks a little bit older? And so to answer that question, she used a machine learning algorithm that right now it's all the rage, a lot of people are using, that essentially predicts your brain age. So all you have to do, you get your structure MRI, you put it into this machine learning algorithm, and the algorithm spits out a number. So your brain looks like you're 25. Maybe my brain looks like I'm 90. I don't know. Okay. And you can see, if you look at the relation between actual chronological age and predicted brain age, these are all patients with back pain, by the way, right? So in the patients that were scanned prior to the pandemic, I think that, the algorithm is not perfect, but it's a pretty good job, right? So you can see subjects that are in their 20s were estimated to be around 20, 40, around 40. Later on, it breaks down a little bit. Also, this has to do with the type of the training data set that was used to train this machine learning algorithm. But you can kind of see that it does overall a decent job. And if you calculate the brain age delta, so the difference between your real age and the age the algorithm thinks your brain looks like, you can see the average is 0. Okay. Now, what happens if we start doing the same, playing the same game with the patient that was scanned after the pandemic? You can see the slope increases, right? You can see that on average, the algorithm estimated the brain age of our participant to be about 10 years older than their real age. This was a largely cross-sectional study, so we cannot, make too much inference about causality, but it was an interesting, nonetheless, and pretty scary proposition, I think. But most importantly, we also found that there was an association between the TSPO PET signal and this brain age delta. So the patients that have higher PET signal were the one for whom the brain looks older than their chronological age. Any questions at this point? All right. So far, I realize I think I have a few more minutes. Yes. So far, I've shown you data from chronic back pain patients looking on the brain. What about elsewhere in their body, right? Do we see that chronic back pain patient demonstrate TSPO PET signal outside of the brain? And for, of course, my colleagues who do preclinical studies looking at microglia and astrocytes, they will tell me, yes, of course, that's what we are seeing, that's where we are studying neuroinflammatory response. There's a lot of focus on spinal cord, for instance, but do we see it in humans? As you guys know, us brain imaging people struggle a little bit with imaging the spinal cord. It's not as easy as we would like, for a couple of reasons we can discuss. But in an interesting study we did in collaboration with Dr. Yi Zhang, very talented pain physician at Mass General, we were able to show that patient with the sciatica, again, these are patients with back pain and leg involvement. This radiculopathy, that's how we call it. We saw elevation in TSPO PET signal in at the level of the spinal cord, especially at the level of the lower level of the spinal cord. These are the segments of the spinal cord that are connected to the roots that are affected by sciatic L4, L5, L5S1. Okay, and we even went further. We started looking even closer to, more and more. We're moving progressively towards the periphery, right? We even saw interesting results at the level of the neuroforamen. Neuroforamen is the part of the spine that contains the dorsal root ganglia and the nerve roots. We cannot really distinguish this structure because of the coarseness of the PET signal. But we saw that on average, there was an elevation in the PET signal even there. So this suggests that TSPO PET signal elevation cannot be seen only in the brain, but also elsewhere in the body, down the spinal cord up to the nerve roots. But are the chronic back pain patients the only patients that demonstrate this TSPO signal elevation? So were we just lucky that we have selected the only patient population that demonstrate this? And the answer is obviously no. We saw elevation in the brain in patient with the fibromyalgia. This was a collaboration with Eva Kosek at the Karolinska Institute, and I was excited. The results were very similar in our cohort here in Boston and in Eva's cohort in Stockholm. But on top of it, they were also largely replicated by 2 additional publications that came after. So this also seemed to be an observation that is quite solid. We found association with the amount of fatigue, excessive fatigue that these patients were demonstrating. Again, a relationship that was observed both at the Boston and the Stockholm cohorts. We did see elevation in the brain of patient with migraine. This is Nushin Ajikani's brainchild, and with relationship with the number of migraines per month. And more and more, we did similar study in gut for illness. Others have done study in CRPS. We're working on HIV neuropathic pain. We're doing pain for trigeminal neuropathy. So really, we're trying to have a sense of how widespread this observation is. And I think that it's fair to say that in our hands, TSPO PET signal elevation as a marker of neuroinflammation seems to be a quite pervasive phenomenon, can be observed in many different ideologically different conditions. So I'm going to add, has to be probably important. We don't know, but it's the gut feeling we have. Now, the interesting point, so while we are seeing TSPO PET signal like, overall elevated in all these conditions, this is not to say that this phenomenon is very aspecific, right? There is some amount of specificity. So for instance, and remember, actually, I should show you the image before, right? So in S1, sorry, for instance, if you look at the fibromyalgia study, you'll see that this is very much widespread signal elevation all over the cortex. Very little thalamic signal there, by the way. And this is very different from what we saw in the back pain. So back pain, we saw a lot of thalamic signal, very little cortical involvement. So this data raises the intriguing possibility that there might be some kind of neuroinflammatory signature that might distinguish different chronic pain conditions, and we're working on that. And of course, this has to be tested more formally. But I find it interesting also to look at the variability that we see in the involvement S1 in different studies, which also suggests there might be some kind of a disease-specific signature. So for instance, if you look at the lumbar radiculopathy study, we saw that TSPO PET signal elevation in S1 was at the level of the back and leg area, right? If you remember your anatomy 101, the Penfield homunculus, you see this part here, right? And how do we know that? Well, this is because this is the PET signal elevation we saw, and this is exactly corresponding to the region that we saw activated using functional MRI stimulation of back and leg. And of course, this is interesting because those are the regions that the participants, where the participant experience clinical pain, right? What about migraine? Well, migraine in S1, we see signal elevation in a much more ventrolateral aspect of the primary sensory cortex in an area that is compatible with the face area. And of course, face is the region that is clinically relevant for them. What about fibromyalgia? Fibromyalgia is characterized by really very widespread pain, often occupying the entire body, and you can see the whole primary somatosensory cortex, right? So this suggests essentially that, although speculative, but it's interesting to potentially speculate that TSPO PET signal elevation might, at least at the level of the primary somatosensory cortex, might have a somatotopic representation paralleling the body distribution of the clinical pain. And again, this has implication for the development of potential biomarkers. Okay, so after having shown you that TSPO PET signal is elevating all these different conditions and seems to have some disease specificity, one question you might ask is, well, does TSPO PET signal change with treatment? And I can promise you we're looking hard. We have many different clinical trials with pharmacological, non-pharmacological challenges. We have mind-body. We have many different way of addressing surgery, et cetera, et cetera. So at this point, I don't have, unfortunately, a lot of answers for you. I'm going to just show you some of the data. So this is the minocycline clinical trial we have completed, which is largely negative. So this is minocycline. You might know it as a tetracycline antibiotic. Is used for acne and bacterial infections. But in animal models, is the probably the drug that's most commonly used as a glial inhibitor. So we're wondering, well, we saw all this signal in the PET signal in the back pain patients. Can we use minocycline to dampen the signal? And does it do anything to pain? And the answer was no. So you can see both pre and post. You can still see this hot spot in the thalamus. Nothing. It was really a beautiful flat line. And there was no effect on pain ratings, right? Both groups go down similarly. Probably this is a regression to the mean or a placebo effect, etc. Now, something that's a little bit, very preliminary, but it's a little bit more intriguing is this N=1 observation we have on a patient with carpal tunnel syndrome. You can see right away this hot spot that correspond, and I promise you have to trust me on this, but correspond to the hand area contralateral to the symptomatic hand in these patients. We were able to rescan that patient after carpal tunnel release, and that hot spot disappeared. Of course, N=1, super preliminary, but was enough, exciting enough for my colleague Vitaly Napadov and myself to decide to do a full study. So right now, if you see a lot of our names in the base 7 schedule, you know why we're doing all these studies, trying to figure out if this really happens in a larger sample. The other question that we were interested in asking is, does TSPO PET signal predict treatment response? And again, this is the paper that I was mentioning before with the lumbar radiculopathy that we did in collaboration with Yi Zhang. And remember, I was telling you that patients with lumbar radiculopathy show this hot spot at the level of the neuroforamina ipsilateral to the symptomatic leg. Well, true, on average, this is true, but there's also variability, right? Some people show a lot of signals, some people show a little signal. And what we're able to show was that patients that did show that signal were the one that responded to epidural steroid injection. It makes sense, right? There's a lot of inflammation there. You use a local treatment that reduces inflammation, or start to reduce inflammation, and that's beneficial. And those patients exhibit 90% of plus relief. If you didn't see any difference between the two sides, the two neuroforamina, well, those are the patients that experience 0% relief. So small, tiny study, but nonetheless, I think quite exciting for us. Then, another study where we're trying to tackle the question, does TSPO PET signal, in this case in the brain, predict response to treatment, was a study that we are writing up right now. So this is in patients with knee osteoarthritis, scheduled to receive total knee replacement. So we scanned them, and we saw, again, high PET signal all over the place, but also using a machine learning algorithm, we were able to predict the amount of relief they experience over time 1 year after the surgery. So again, to me, what this is exciting, the reason why this is exciting, because it suggests that what we're showing is not just an epiphenomenon, but might have something, the TSPO PET signal elevation might be relevant for the establishment and maintenance of persistent pain, or development of persistent post-surgical pain in this case. Let's see how much time do I have. Any question at this point? Yes.
Well, it's like the
I wasn't sure I could understand. I could. No, no, it's a very good question. So what are the mechanisms leading to this upregulation of TSPO? Interestingly, we don't know a lot of it. So let me just get back to you about this one, right? But essentially, TSPO seems to be upregulated. One of the hypotheses is that TSPO is upregulated as a negative regulator of inflammation, almost like as a means to dampen the inflammatory responses after they have been initiated. I mean, people are figuring out all the, the biochemical cascades, etc. But I can tell that there is really a lot that is unknown at this point. We call it translocator protein because it's, for instance, involved in the transfer of cholesterol molecule across mitochondrial membrane, which is thought to be, which is the rate limiting step for neurosteroidogenesis. How does this link to the inflammatory responses is something we are trying to figure it out. Now, I promised you I was going to go away. Oh, yes.
Well, along those lines, I was wondering, there's a bunch of older studies that show that acute inflammation can increase glucose. So there's a lot of other markers one can pick besides just looking at alterations of pathology specifically. Just wondering if you guys have done.
No. So yeah, no, you're right. Absolutely. There are other tools that people can use. And the entire field is actually trying to aggressively look for additional targets because, there's question about specificity. And I'll get back to you in 2 slides about that. Okay. And in fact, well, the point that I'm making, so one of the concerns that people have is that, this lack of specificity is, you can see is expressed in a different cell type. You're not sure what you're exactly imaging, right, at this point, right? So is it microglial cells, it's astrocytes, and about this, what I have to say is this, that first of all, at least in the context of pain, we are still at a stage where we still don't know to what extent brain inflammation, neuroinflammation even takes place. So a target is a little bit broader, right? Doesn't maybe give you the specific resolution of the specific cell types that's driving it, but it's a marker of broad inflammation, still very valuable. And the other thing I want to say is, I think I'm going to make myself this nice segue to the next slides I want to present, is that the downside of lower specificity is versatility. And this is why, because of this versatility that TSPO has, it's expressed not only centrally, but also expressed, for instance, in the periphery, that we can use it as a potential marker to image central, but also peripheral inflammation. And we tested this hypothesis in knee osteoarthritis, and we were able to show that in patients with knee osteoarthritis, we were imaging, of course, focused on the brain, but in the end we said, well, why don't we also image their joints? Right? It makes sense. TSPO is also expressed by, monocyte, macrophage, fibroblast. There's a bunch of different cells that make sense to image. And so indeed, we were able to see that in patients with knee osteoarthritis, so TSPO signal elevation in the joints, bilaterally in the patient that have bilateral inflammation. These are representative patients. No, that was a joke, sorry. But unilateral in the patients with unilateral knee osteoarthritis. These are the statistics here. And we saw correlation with pain. We have this collaboration with Mohammed Jaraya, who is a musculoskeletal radiologist. He reviewed all the, was that MRI from this patient? Radiographs, and basically did all the scoring according to the, clinically used scales, the Kellgren-Lawrence scale, and was able to show that, looking at, the amount of osteocytes or the degeneration of the cartilage, etc., did not really tell us how much pain the patients had. This is the PET signal was a better predictor. We also saw association with the cytokine levels in a small subsample of the participant from whom we collected those data, TNF-alpha, interleukin-1 RA, et cetera, suggesting that what we're really imaging is related to inflammation. In summary, TSPO signaling is elevated in multiple etiologically different chronic pain condition, but it seems to exhibit a degree of disorder specificity needs to be tested more formally than the way I showed it. It appears the clinical significance of the signal depends on the brain region. If Katerina shows me a TSPO PET signal in a region on one of her patients, I'm not going to say, well, that's pain because I showed TSPO. It depends on the pathology and depends on the region, right? It seems to be that it can predict treatment outcome, at least in the study that I showed you. It may respond to treatment, although definitely more work is needed. And I'm going to argue it's also a promising marker of peripheral inflammation. I think I'm done. There's no time to talk about everything. I'll just mention the future direction. See if I have 1 minute. Yes, future direction. Bruce, you were mentioning about other tools. Definitely, there are other tools that people are trying to develop to image inflammation with more specificity. Some of those actually developed here. Talk to Jacob if you're interested in this COX-2 ligand. There are other ligands. Definitely a very, very dynamic and exciting field. There are also methods that we and other people are trying to evaluate to assess the presence of neuroinflammation based on, for instance, diffusion. So we are doing this study in Bay 8 to essentially try to take advantage of the diffusion properties of water molecule to try to see if we can detect those cell swelling, this increased cellularity, this retraction of the processes, all these histological changes that we know happen in neuroinflammation. We're able to see it in the connectome scanner, and this is work that's been published recently. So we're trying to test it out. And also, in addition, we are often ourselves and other people use spectroscopy, another tool to collect measures that we think they're related to inflammation, right? For instance, in spectroscopy, you might look for the concentration of myonocytes. Some people say, well, maybe that's a glial metabolite, so that's evidence of inflammation. Well, now we're in a position to validate these tools, right? Can we use PET MRI, against one of these radioligands or maybe TSPO ligands to really give support to the notion that, I don't know, myonocyte or choline, those are inflammatory markers. And I think this would be very exciting, because if we could do without PET in terms of, how expensive, how, tricky it is to run this experiment, I think it would be advantageous from the standpoint of the clinical translation of our observation. And I want to thank, really the lab. I really very lucky to work with a lot of wonderful people. Martinos Center is an amazing place. Definitely, we couldn't have done this stuff elsewhere in the world. The funders and of course, all the participants of our studies, they are asked to do a lot, I have to say, and they're wonderful. And of course, all of you for attending this talk. Thank you, guys.